In Chapter 6, we will be discussing and reviewing recombinant DNA. We'll talk about what is recombinant DNA, how it's prepared, and also talk about cloning of recombinant DNA, and then the uses of the cloned recombinant DNA for gene therapy as well as generation of transgenic animals. In addition, we'll talk about how we prepare recombinant proteins from the recombinant DNA. Now, in this first figure is an overview of cloning of recombinant DNA. In my opinion, don't go drawing down details. This is just an overview. The details will come later on in this chapter. Just an overview of what recombinant DNA is and how it's cloned. Now, what is the definition of recombinant DNA? Recombinant DNA, by definition, is a splicing together of DNA from two sources. And the two sources most often is some sort of human DNA and spliced together with a bacterial vector. It does not have to be human DNA. It can be chimpanzee DNA or something like that. And it can be spliced into some sort of bacterial vector. And again, it doesn't have to be a bacterial vector. It could be a yeast vector. But on the USMLEs, if they generally talk about recombinant DNA, it'll be in the context of human DNA inserted in some sort of bacterial vector. Now, the first issue we want to discuss in terms of this overview is, where does the human DNA come from? What's the source of human DNA? Well, one of the two sources of human DNA is genomic DNA. Simply put, if you take some of your tissue and put it in a test tube, get rid of all the membranes and get rid of all the histones and the chromosomes, you have naked DNA. And if you take the naked DNA from all 46 chromosomes, add some sort of endonuclease, you'll fragment the 46 chromosomes into a multitude of DNA fragments. And that's called genomic DNA, fragments from the entire genome of a particular individual. And all of those will be small amounts, and they'll be very heterogeneous because it's fragments from all 46 chromosomes. Another type of source of DNA for the preparation of recombinant DNA, it's what's known as cDNA. cDNA simply means is some tissue is expressing genes into messenger RNA. Then the messenger RNA, by reverse transcriptase, is converted to cDNAs, complementary DNAs. Again, the details are coming later. Don't go worrying about them yet. But these will be complementary DNAs to the messenger RNAs that are being expressed by a particular tissue. Again, there will be very small amounts of those cDNAs because they're small amounts of messenger RNAs, and in addition, they're very heterogeneous because each tissue is making lots and lots of different kinds of messenger RNAs. But what is the goal here? The goal is to amplify a single piece of DNA from genomic DNA, or to amplify a single cDNA amongst all the other cDNAs. We want to amplify a single piece of DNA or a single cDNA. Now, the way this is done, again in this overview, is to take the genomic DNA fragments or the mixture of cDNAs. These are then introduced into plasmids, and now we end up with recombinant plasmids, where human DNA pieces or cDNAs are inserted into a bacterial vector. So these are recombinant plasmids. And then these recombinant plasmids are then added to bacteria, and in a process called transformation or transfection, the bacteria will take up these recombinant plasmids, and then the bacteria will continue to grow. And at this stage here, we end up with a mixture of bacteria containing different recombinant plasmids. So in a test tube or a flask, we have a mixture of bacteria. Each bacteria has a different recombinant plasmid that they're harboring. Now, in microbiology, you probably all have done this. In microbiology in the lab, you've taken some sort of bacterial mixture and taken that mixture and streaked it onto soft agar dishes. And in anywhere where a single cell sat down on a soft agar dish, what will happen is a colony now will appear. So what this represents here is a agar dish where a single cell sat down on an agar dish and formed a colony. Importantly, at this stage here, each bacterial colony harbors a different recombinant plasmid. And so at this stage here, each of these colonies is actually a clone of the original colony. And so this entire plate is what's viewed as a DNA library. So if way back at the beginning, if the source of DNA is genomic DNA, then this library can now be called a genomic DNA library. If the source of the DNA is cDNA, then this library now would be called a cDNA library. But importantly, each of these colonies is a clone. And so what's done now is to take a particular clone. Let's assume we're going to take this particular clone right here that's harboring a specific gene of interest, for example. Let's assume it's harboring a single gene of interest. So we take this colony and then take the colony and put it into some flask which has medium, growth medium. So we take the colony, pick the colony and put it into growth medium and now grow this particular colony in this growth medium. What can we do with this colony now that's growing? This is E. coli or whatever bacteria of interest. In this case here will be E. coli in this example that's now harboring a particular plasmid, harboring a particular gene of interest. Now if we grow this up, what can we do with this particular colony that's growing up in medium? Well, one thing could be done would be to induce the gene expression, and now this E. coli is now taking the recombinant gene and now making it into recombinant protein. And so this protein can now be used for treatment of particular diseases. For example, if this is a colony that harbors the insulin gene, then the insulin gene can now be making insulin protein, and that protein now can be used to treat diabetes, for example. What's another thing that can be done with the colony that's now growing in this growth medium? Well, what can be done is to take the bacteria and lyse the bacteria with some sort of detergent. So that now the bacteria are then lysed and then all of the recombinant plasmids spill out. We can take the recombinant plasmids and then get rid of the bacterial vector portion and now take the gene that was in that recombinant plasmid, and now we have amplified that particular DNA. So again, way back at the beginning, the goal was to amplify a single piece of DNA. If we take the recombinant plasmid, which has now been amplified by the bacteria, we have now at this stage amplified that piece of DNA or that cDNA that we were interested in. So we have now cloned that particular DNA. We have made mega quantities of either a piece of DNA from genomic DNA or cDNA, because way back at the beginning that was the source of the DNA. At this stage here, that DNA we have now made in large amounts and it's now homogeneous. Because why? Because way back at the agar dish stage, we have a colony which harbors a different recombinant plasmid. So now at this stage over here, that DNA of interest is now homogeneous. The issue now becomes, well, what are you going to do with this DNA now that we have large amounts of homogeneous DNA? Well, the uses of that DNA that we'll be talking about in Chapter 6 in greater detail later on. One use of that cloned DNA is for gene therapy, how that gene can be used to treat and to cure a defective tissue. The other use of that cloned DNA is involved in the preparation of transgenic animals. And these transgenic animals are models for analyzing human diseases. And the details, again, will be coming up later on in Chapter 6. On this figure here is a discussion of how a genomic DNA library is prepared. So what we have here at the top of this figure is a portion of a chromosome. Let's assume in a portion of this chromosome, here's a gene, and just upstream of the gene is the promoter, P standing for promoter here. And just upstream from that is the enhancer for that particular gene. Now, in the vicinity of this particular gene, there are palindromes for a particular endonuclease. Now, what is a palindrome? A discussion of what a palindrome is will come on the next figure. But palindromes are simply a DNA sequence which is cleaved by a particular restriction endonuclease. Again, a discussion of this comes on the next figure. But in the vicinity of this gene, these vertical lines represents palindromes for a particular endonuclease. So if we take a chromosome, chromosomes, and add a restriction endonuclease, the endonuclease will look for its palindromes, and in this example here, it'll cleave at this side here, 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 and here, and generating what's known as DNA restriction fragments. Notice how we get a very large DNA fragment and some smaller DNA fragments. And in fact, one of the palindromes was in the particular gene here, and notice how the gene itself got fragmented. But importantly, we have now generated a number of DNA fragments. These DNA fragments can then be ligated into vectors. Now, ligation means simply splicing of two segments of DNA by reforming phosphodiester bonds. And then these fragments are spliced into vectors. We have now generated a series of recombinant vectors. These are then transformed into bacteria. The bacteria are then cloned on soft agar dishes, as was discussed on the previous figure. And then what we have now generated is that in this plate are all these colonies, and the total bacterial colonies is now called a genomic DNA library. Importantly, each colony contains a different fragment from the entire genome from that particular organism. On that plate, each colony has a different fragment from all 46 chromosomes. So if you added up each of the fragments, that should be the entire 46 chromosomes. Okay, so this is how a genomic DNA library is prepared. Next discussion deals with restriction endonucleases. Now what endonucleases are is, I just would like to do first is to compare that with an exonuclease. What an exonuclease is by definition, an exonuclease works at one end and cuts phosphodiester bonds to release single nucleotides. Now in biochemistry so far, in the previous chapters, we have already talked about two important exonucleases. We talked about a 5' exonuclease and we talked about a 3' exonuclease. So obviously I'm going to ask the question, what was the 5' exonuclease? The 5' exonuclease was the enzyme which removes an RNA primer during replication of DNA. So the RNA primer is removed by the 5' exonuclease and then replaced with DNA. What was the 3' exonuclease that we talked about in the previous chapter? What the 3' exonuclease is, it's an enzyme within the DNA polymerase, which when the DNA polymerase pauses and checks and finds out it makes a mistake, the 3' exonuclease within the DNA polymerase will take out the mispaired nucleotide at the 3' end and replace it with the correct nucleotide. So 5' and 3' exonucleases are very important. Now how does this contrast with endonucleases, restriction endonucleases? What an endonuclease is by definition, it's an enzyme which cleaves internal phosphodiester bonds and releases restriction DNA fragments. I want to emphasize this is not at random. It does not cleave phosphodiester bonds at random. What it first must need to see is a particular DNA sequence. And a DNA sequence that a particular endonuclease is looking for is viewed as being a palindrome. Now what is a palindrome? A palindrome is a DNA sequence which reads the same backwards and forwards. Generally, palindromes are 4 or 6 or 8 base pairs in length. There are exceptions to that, but on the USMLEs, it'll be an even number, either 4, 6, or 8 base pairs in length. Now, shown on this figure is an example of a palindrome. Here's a palindrome, 5' GAATTC. Now, you're going to look at that and say, you know, that's not a palindrome, because palindromes, when you say reads the same backwards and forwards, like the word Xanax. Xanax is a palindrome. It reads the same backwards and forwards. XANAX reads the same backwards and forwards. That's a palindrome. But here, a palindrome regarding DNA is that it reads the same backwards and forwards on the opposite strands of the DNA, not on the same strand of the DNA. Because here, it's on one strand it's 5' GAATTC to the 3' end. Now on the opposite strand, it reads 5' GAATTC to the 3' end. So a palindrome of DNA is where it reads the same backwards and forwards on the opposite strands of the DNA. And then once a palindrome is seen by a particular endonuclease, it will then cleave at within that palindrome. Now, on the national board, they might ask a question about palindromes, and they might ask you to identify a palindrome, and the issue becomes, is there an easy way to determine if something is a palindrome without wasting valuable time, valuable seconds, trying to figure out if it reads the same backwards or forwards on the opposite strands of the DNA? For example, they might give you this double-stranded DNA as you see here and ask you, is that a palindrome? Or they might give you one strand of DNA and ask you, is that a palindrome? For example, here it is right here. Here, if they give you a single strand of DNA and they say to you, is this a palindrome? Now you can sit there and waste valuable seconds by drawing out the other strand and then trying to figure out is it reads the same backwards and forwards, but there's indeed an easier way to determine that a single strand is a palindrome or not. Now, the way you do this is you look at that single strand, and I'm going to use my cord here. If you take a single strand of DNA, what you got to do is that in your mind, bend the DNA in half like this. When you bend it in half like that, ask yourself the question, does that nucleotide on one side base pair with the one on the opposite side? If the answer is yes, you go on to the second nucleotide. Does that nucleotide on one side base pair with the one on the opposite side? If the answer is yes, you go to the third nucleotide. Does that nucleotide on one side base pair with the one on the opposite side? If the answer is yes, that is a palindrome. So now, look at this particular sequence here, GAA-TTC. In your mind, bend it in half. When you bend it in half, does that A base pair with that T? The answer is yes. You go to the second nucleotide. Does that A base pair with a T? The answer is yes. You go to the third nucleotide. Does that G base pair with the C? Their answer is yes. Then that is a palindrome. In fact, you can do that with either strand of the DNA. Look at the bottom strand. In your mind, bend it in half. Does that T base pair with the A? The answer is yes. Does that T base pair with the A? The answer is yes. Does that C base pair with the G? The answer is yes. So even if they gave you this strand of DNA, then you would say, yes, it is a palindrome. And then as a consequence, what would happen is, this is the particular palindrome for the endonuclease called EcoR1, and EcoR1, when it sees its palindrome GAA-TTC, it will cleave, as shown by the arrows, between the G and the A of one strand and the G and the A of the other strand. We'll talk more about EcoR1 on the next figure. And so it will cleave at this particular palindrome. So how might they ask about palindromes on the USMLEs? There's no way they're going to ask you about particular sequences. You don't have to memorize the sequence for EcoR1 which is being recognized. How they might ask this on the national board is shown here. They might ask you, they might make a statement. The restriction endonuclease, called PST1, cleaves a double-stranded DNA at a site beginning with CTG. What's the remaining sequence? And then they give you 5 options. So as you know, most palindromes are even number. So you would say, well, if you draw out CTG, here's CTG, then the answer is going to have 3 nucleotides as an answer, because that will give 3 plus this 3 here that they start with will be 6. So in your mind, bend the CTG in half. The G base pairs with a C, the T base pairs with the A, and the C base pairs with a G. And now this is the remaining sequence, which is seen by the restriction endonuclease, PST1. So this is how they could do this on the national board exams. Now, coming back to this one over here, the GAA-TTC, this is the palindrome seen by EcoR1, which is a very commonly used endonuclease. It's going to cleave between the G and the A and the G and the A. Now, this is going to be shown a little bit better on the next figure. So here is that same sequence, GAA-TTC on one strand. On the opposite strand, it's 5' GAA-TTC. And then when EcoR1 sees it, it cleaves between the G and the A of that strand and the G and the A of that strand. And notice how this is the fragments being generated from this particular palindrome. And importantly, notice how those fragments have single-stranded domains. And this is called sticky ends. TTAA, this is a single-stranded domain of this double-stranded DNA fragment. And here of another DNA fragment, here's another sticky end, which is AATT in this example. So with sticky ends of fragments, of DNA fragments, these are overhangs. So a sticky end is a single-stranded domain of a double-stranded DNA fragment. And these are widely used in cloning, which is coming up very shortly, and we'll be reviewing that soon. Now, there are other kinds of endonucleases out there which are not used that much in cloning. For example, here's one on the right side of this figure called HA3. The palindrome for HA3 is a sequence of GGCC. Now, if you look at this very quickly, you can determine that it is a palindrome by bending it in half. Notice how the G base pairs with a C, the G base pairs with a C, and notice how quickly you have determined that this is a palindrome. In fact, it's the palindrome for the endonuclease HA3. But when HA3 sees its palindrome, what it does, it cleaves right down the middle and generating no sticky ends, and it's called blunt ends. There is no single-stranded domains, and as you'll see shortly, this is why it's not used in cloning techniques. But there are examples of endonucleases which do not generate sticky ends, and this one here is HA3. So where we're going next is to talk about how recombinant plasmids are formed. So on the upper portion of this figure represents, let's assume, some human DNA. What this represents is a portion. This represents a portion of one chromosome among 40 chromosomes. Now at this portion of the chromosome, there are palindromes. So here's a palindrome again, GAA-TTC. This is the palindrome for this enzyme called EcoR1. Now what would happen if we take 46 chromosomes and put them in a test tube and add EcoR1? What's going to happen is this EcoR1 will look for its palindrome GAA-TTC and generate sticky ends. So when this chromosome is seen by EcoR1, it's going to cleave it, generating sticky ends. So what you see here is this fragment coming from a particular chromosome, generating the sticky ends, AATTC. Because why? Because the palindrome is being cleaved between the G and the A, and then on this side over here, it's cleaving between the G and the A there. So now we generated a fragment, a double-stranded DNA fragment with sticky ends. But keep in mind, this is one fragment among hundreds, if not thousands of fragments, because if you have 46 chromosomes in a test tube, you add EcoR1, anywhere it sees its palindrome, it's going to cleave it. And all 46 chromosomes are going to have a multitude of palindromes. So this represents one fragment among many fragments that are being generated when 46 chromosomes are seen by EcoR1. Okay, so this is being done in one test tube. Now in another test tube, let's assume we put in some sort of bacterial plasmid or bacterial vector. Now this is given a specific name here. Don't go worrying about a name. Just know it as a generic bacterial vector or bacterial plasmid. Now this vector will have the same palindrome, GAA-TTC. So this is a plasmid that's sitting in a test tube. What would happen if we add EcoR1 into that test tube? What's going to happen is the EcoR1 will look for its palindrome and cut it, as you see here, generating sticky ends on the plasmid. And now we have two test tubes. In one test tube, we have plasmids which were cut with EcoR1. In the other test tube, we have chromosomes which were fragmented by the same EcoR1, hundreds if not thousands of fragments. Now what's going to happen when we mix these two test tubes together? What's going to happen is each of these fragments, for example, this fragment here, is now just like a cassette in a tape player. This fragment now will insert into this area right here of the cut vector. And notice how this AATT there will base pair with the TTAA there. And similarly, this TTAA of this fragment will base pair with the AATT of the bacterial vector. So like a cassette in a tape player, this fragment now inserts itself right into this portion of the cut DNA vector from the plasmid. And then if we add DNA ligase to reform the phosphodiester bonds, we have now generated recombinant DNA, where a human DNA fragment has now been inserted into a bacterial plasmid, a bacterial vector. Now to illustrate this a little bit better, if this portion over here, this DNA fragment, is now shown in color with the magenta, in this recombinant DNA, it now will appear here. So this is a human DNA insert in some sort of bacterial vector. Keep in mind, this is one vector containing a single DNA fragment. So each bacterial plasmid contains a different human DNA fragment. But in this test tube, there's not going to be just one plasmid with one DNA insert. There's going to be hundreds, if not thousands, of plasmids, and each plasmid, each vector, has a different DNA, human DNA insert. Okay, so the next discussion and review has to do with the bacterial vector. There's some issues that we need to review regarding the bacterial vector or the bacterial plasmid. But what we got on this figure is the cloning, how we generate a cDNA library. Earlier we had discussed how genomic DNA libraries are prepared. On this figure is how cDNA libraries are prepared. So here we have, in this figure, if we have a tissue which has genes that are being expressed. So these genes would be in euchromatin. These genes, as you know, are DNA, and these genes are being expressed into messenger RNAs. Now, importantly, these messenger RNAs, when they are being used to generate a cDNA library, are being isolated from the cytoplasm of tissue, not from the nucleus of tissue. They're coming from the cytoplasm of tissue. So these are cytoplasmic messenger RNAs. So it brings up the issue, why is it so important that these are cytoplasmic RNAs and not nuclear RNAs? And the reason for this comes back to the previous lectures that we had in the earlier chapters in biochemistry. Remember that when DNA is being expressed, it's being expressed into a primary RNA transcript, and the primary RNA transcript has to be processed three ways. First, there's a 7-methyl G cap that's added co-transcriptionally, then it's called hnRNAs, and then a second type of transcriptional processing event occurs in which a poly-A tail is added, and that's done post-transcriptionally. And thirdly, and most importantly, all these introns that genes have, which are in the primary transcript, and then subsequently in the hnRNA, these introns are spliced out. So mature messenger RNA is being generated in the nucleus, and then when the messenger RNA is mature, after all three transcriptional processing events occur, then the messenger RNA gets out into the cytoplasm. So now, in the cytoplasm, we have a mixture of messenger RNAs. These are all mature. The processing events have been completed, and importantly, these messenger RNAs have no introns. These messenger RNAs have the complete reading frame in order to encode for proteins in translation. Now here we have these messenger RNAs which can then be isolated. They can be put into a test tube, and then if a reverse transcriptase is then added to the test tube, reverse transcriptase will take the messenger RNAs and make a mixture of cDNAs. Now what does a C stand for in cDNAs? It stands for complementary to the RNA. So these cDNAs are complementary in sequence to the messenger RNAs. So at this stage here, we would have a mixture of cDNAs. Why a mixture? Because whatever the tissue is, there's a mixture of messenger RNAs that are being generated, which are then converted by reverse transcriptase to a mixture of cDNAs. What happens next is this mixture of cDNAs is then ligated into vectors, again spliced into vectors using DNA ligase to reform phosphodiester bonds, and then these recombinant plasmids are then transformed into bacteria, and then when the bacteria are then grown up and cloned on soft agar dishes, the total bacterial colonies at this stage here now represent an expression library, more commonly called a cDNA library. So on a national board, you might see it as a cDNA library, or you might see it as an expression library. Where does the word expression come from? Way back at the beginning. These genes are being expressed into messenger RNAs, which are converted to cDNAs. So an expression library or a cDNA library refer to the same thing. Now on this agar dish with each of these colonies, which has a different cDNA, each colony contains a cDNA from that particular tissue that we started with way back at the beginning. Okay, so this is how cDNA libraries are generated. So now what we want to do here is compare. This is a very important table that compares genomic libraries and cDNA libraries. So there's nothing new here, it's just comparing the two types of libraries. The first thing here is, at the top of this table, is the source of the DNA. For the generation of a genomic library, we start off with chromosomal DNA. Where did it come from? It came from the nucleus, because the nucleus is where the chromosomes are located, and then we can then fragment them by endonucleases. But cDNA libraries originated, the source of the DNA came originally from messenger RNA. And where did that come from? It came from the cytoplasm of some tissue that's expressing messenger RNAs. Now the enzymes to make the library, to make a genomic library, what's needed is a restriction endonuclease. Why? Because we have 46 chromosomes. They are then fragmented with a particular endonuclease. And then a DNA ligase is necessary to take the individual DNA fragments and then splice them with some sort of bacterial vector. Now with preparation of cDNA libraries, there are no endonucleases required. The enzyme that's required there is reverse transcriptase, and the reason for that is to take the messenger RNAs and convert the messenger RNAs to cDNAs. And then DNA ligase is necessary to take the cDNA and splice it into some sort of bacterial vector. Now continuing on, it says one of the items is it contains non-expressed sequences of chromosomes. With genomic libraries, the answer is yes. Why? Because in genomic libraries, there'll be introns as well as exons. As you know, introns are not expressed. In addition, there'll be spacer DNAs. So those spacer DNA is not expressed downstream into protein. So the bottom line to a genomic library, the reason why it's yes is because it contains all the sequences from a particular genome. Yes, the genome has been fragmented, but all the fragments should add up to all 46 chromosomes. Now with the cDNA library, the answer is no here. And the reason why it's no is because a cDNA library corresponds to genes that have been expressed into messenger RNA into that particular tissue. Once the messenger RNA is generated, reverse transcriptase converts it to cDNA. So it does correspond to genes expressed in a tissue. So all of the RNA has been expressed. Another item contains genes, clone genes are complete sequences. And it says here, not necessarily with genomic libraries. Why not necessarily? And the answer to that is, remember in one of the earlier slides how we had a particular gene in generation of genomic libraries, how a gene might have a palindrome within the gene. When we add an endonuclease to a chromosome which has a palindrome within a gene, what's going to happen is that gene might be cut up if there is a palindrome within the gene. So a genomic library will not have complete sequences of particular genes. But with a cDNA library, the answer is yes. Why? Because there are no endonucleases that are being used to fragment nucleic acids. We have complete reading frames of messenger RNAs, which are converted to cDNA. So it's the complete sequence. That's why the answer is yes. Another item. Clone genes contain introns. Certainly with cDNAs, the answer is yes, as we just talked about up here. With cDNA libraries, there is no introns. Why? Because the introns have been removed during the processing of primary RNA transcript into mature messenger RNAs. What about promoter and enhancer sequences? Are they present? In genomic DNAs, the answer is yes, but with cDNAs, the answer is no. Again, why is that no for cDNAs? And the answer to that is during transcription, promoter sequences, enhancer sequences are never transcribed into RNA. They are used, but not transcribed into RNA. So that's why the answer is no with cDNA libraries. What about the genes that are in genomic libraries? Can they be used in generation of recombinant proteins? The answer is no for genomic libraries. And why is that? The reason for that is the genes way back at the beginning may have had a palindrome within the gene, and the gene then is fragmented. And so therefore, if you have a fragmented gene, obviously you can't make a mature recombinant protein downstream. But with the cDNA library, the answer is yes, because messenger RNAs have a complete reading frame for translation of proteins, and then those are converted to cDNAs. So the answer is yes for cDNA libraries. And then finally, can these libraries be used for gene therapy or for generating transgenic animals? And the answer is no for genomic libraries. Why? Again, the genes that are in the genomic library most likely have been fragmented because genes likely would have palindromes within them, which would be fragmented, cut by endonucleases. But with cDNA libraries, the answer is yes, because the cDNA libraries have a complete reading frame for the translation in order to make proteins. Now, with this information on this table, I got a series of 4 questions to ask you so that hopefully you can understand what's going on here. Suppose that we want to clone the insulin gene. So what I'm going to ask is 4 questions, and it has to do with cloning of the insulin gene. Now, for this context, just assume that the insulin gene has no palindrome within it. But let's assume you wanted to clone the insulin gene that has no palindrome in the insulin gene. Can you use as a source genomic DNA from the pancreas? Is the answer yes or no? If you wanted to clone the gene for insulin, can you go to your pancreas, get your chromosomes, your 46 chromosomes? Do you have the insulin gene present? And the answer is an easy answer. The answer is yes. Why is it yes? Because everybody knows the pancreas makes insulin protein, therefore it has to have the insulin gene. Now the second question is, can you use genomic DNA from the brain? Now, as everybody knows, the brain does not make insulin protein, but does it have the insulin gene present? And the answer is yes. The insulin gene is present in the brain. It's not in euchromatin, it's in heterochromatin. The insulin gene is in every cell in your body except erythrocytes, because erythrocytes don't have nuclei. But it's in every cell in your body. So if you wanted to clone the insulin gene using genomic DNA technology, you can get the insulin gene from any cell in your body, because within the 46 chromosomes, one of the chromosomes, two of the chromosomes, matching chromosomes, will have the insulin gene. Now, another question. Can you use, if you wanted to clone the insulin gene, could you use cDNA from the pancreas? And the answer is yes. The pancreas, as everybody knows, has messenger RNAs for many proteins to encode for many proteins. All the enzymes in pancreatic juice, pancreatic lipase, pancreatic amylase, and so on. But at least one of the genes that's being transcribed in euchromatin in the pancreas is the gene for insulin. So among the multitude of messenger RNAs being present in the cytoplasm of the pancreas, one of the messenger RNAs will be the messenger RNA for insulin. And so if you add reverse transcriptase, the messenger RNA for insulin, as well as the messenger RNAs for the other proteins, will be converted into cDNA. So the answer is yes. Now lastly, if you wanted to clone the insulin gene, could you use cDNA from the brain? And the answer is no. The brain makes many proteins, and therefore the brain has many messenger RNAs, but not one of those messenger RNAs will be the messenger RNA for insulin. And the reason for that is the insulin gene is present, but it's in heterochromatin and it's not being expressed into messenger RNA. So the answer is here, no. Okay, so now this is again a comparison of genomic and cDNA libraries. So now with this, on this figure, we need to discuss several details of the vector itself, the bacterial plasmid or the bacterial vector, same thing. Now it says here this is an expression vector, but in fact there are two kinds of vectors that can be used in cloning. One is an expression vector, as you see here. Another kind of vector that can be used in cloning is what's known as a replication vector. And that's what I want to talk about first. What is a replication vector? As the name implies, this is a sequence of DNA, some bacterial plasmid, some bacterial vector, which when you give it to a bacterium, such as E. coli will take up the vector and replicate it just like its own chromosome. So in order for a piece of DNA, circular DNA, to be classified as a replication vector, there are three criteria that must be met. And the first criteria that must be met in order for a vector to be classified as a replication vector is that there has to be an origin of replication, abbreviated as an ORI site. This is a small sequence of DNA in the circular DNA, which is recognized by the bacterial replicating enzymes. Because after all, replication does not take place at random. The bacterial RNA, bacterial replicating enzymes must see a particular sequence of DNA. When it sees that sequence, called the ORI site, then the enzymes then sit there and then start replicating the DNA. So that's the first criteria that must be met. A sequence must be in the circular DNA, which is recognized by the bacterial replicating enzymes. The second criteria that must be met in order to be a vector to be classified as a replication vector is that there has to be a single site. The vector has to have a single site for a given restriction endonuclease. A single site, not multiple sites. Now, why not multiple palindromes within this particular vector? Now, if you have a circular bacterial vector, what would happen if you had multiple sites, multiple palindromes? So if you had multiple sites, you had some sort of endonuclease. If you have a palindrome here, it would cut here. If there's another palindrome here, it would cut here. Everywhere it sees a palindrome, the endonuclease would fragment the circular DNA. And then when you added your bacterial insert, it would be like trying to put Humpty Dumpty back together again, and you can't do it. There's too many pieces to try to reassemble into some sort of vector. But there has to be a single site for a given endonuclease. If you have a circular DNA with a single palindrome, you add EcoR1 or whatever enzyme we're talking about, you add EcoR1, it would cut the DNA, open it up, but not cut anywhere else. But now that the vector is opened up, we can now add our piece of DNA, human DNA. You can now insert it, add DNA ligase, and now reseal the circular DNA, and we have now inserted our human DNA fragment. That's the second criteria that must be met in order to be classified as a replication vector. The third criteria that must be met in order for a vector to be classified as a replication vector is that there has to be, in this vector, there has to be a gene that encodes for antibiotic resistance. Now, you know from microbiology, a lot of plasmids have genes which will inactivate a particular antibiotic. That's why a lot of bacteria become resistant to penicillin or ampicillin as time goes on, because they can pick up a particular plasmid, and that plasmid might have a gene which inactivates penicillin or ampicillin or some other antibiotic. So this vector will have a gene within it which, once it's made into messenger RNA, will encode for an enzyme which inactivates a particular antibiotic. Now, why is that? Now, the answer to the question why we'll talk about on the next figure once we get to cloning. Why you want a gene present which will inactivate a particular antibiotic. So now, if all three of these criteria are met, this vector, the circular DNA, is now classified as a replication vector. The E. coli will take up the vector and replicate it just like its own chromosome. So as the bacteria grows, it replicates its own chromosome and replicates its vector, and each bacteria now has a particular replication vector within it. But now the important issue is, if we added some human gene at this site here, because we opened up the plasmid, added our human gene there, and sealed it back up with DNA ligase, what will happen is the bacteria will take up this vector and replicate that gene in the vector, so now we have generated tremendous numbers of that particular gene. But now, will we be making large amounts of the human protein from the human gene that has been inserted? And the answer is no in a replication vector. And why not? And the reason being is, yes, there's a human gene present, but there is no promoter that the bacterial RNA polymerase can recognize in this circular DNA. And not only that, even if there was a sequence, a promoter for the gene present, then the messenger RNA is made. No human gene has a Shine-Dalgarno sequence. Now, what's the function of a Shine-Dalgarno sequence from the previous biochemistry lectures? And the answer is, a Shine-Dalgarno sequence is a small sequence of RNA, which comes from the DNA originally, and it's a sequence of RNA to which the bacterial ribosomes need to see in order to jump on the messenger RNA to translate the RNA into proteins. So a replication vector does not have a promoter and does not have a Shine-Dalgarno sequence. But now, if we take a replication vector, which has an origin of replication, a single site for a given endonuclease, and a gene which will encode a particular antibiotic, if we now engineer into this replication vector a sequence for a promoter for a bacterial DNA, RNA polymerase, as well as engineer a Shine-Dalgarno sequence, we can now convert this replication vector into what's now known as an expression vector. So an expression vector, just to summarize, is identical to a replication vector, except now it has two additional criteria that must be met. There has to be a promoter that's engineered near the gene of interest, the human gene that's been inserted, and then what's going to happen is the bacterial RNA polymerase will jump onto the promoter and then start transcribing the gene. That's going to be illustrated here. So the RNA polymerase will start at the 5' end and then go to the end where we get termination. So now we have generated a messenger RNA. Why? Because a promoter has been engineered into this vector. In addition, the fifth criteria is that in this vector we have engineered a Shine-Dalgarno sequence into the DNA. So then when the RNA polymerase then starts transcribing the gene into RNA, the Shine-Dalgarno sequence, which is right next door, just upstream of the human gene, now will be present. Now that's shown here in the green. So the green is representing the Shine-Dalgarno sequence, which has been engineered right next door to the human gene, which is now being made in the human messenger RNA. So at this stage here, this is now viewed as being an expression vector. So to summarize, a replication vector has three criteria, and an expression vector has those same three criteria, but with two additional important features, and that's a sequence for a promoter that the RNA polymerase from the bacteria must recognize, as well as a Shine-Dalgarno sequence. So after this, the next figure to talk about is how we actually do the cloning process. What we have on this figure is a review of the basic principles behind cloning. Now you might say to yourself, why do I need to know about cloning? Because you might say, wow, I'm never going to do cloning in your career. But the USMLE expects you to understand just the basic principles, a basic understanding of what cloning is all about. And the reason is many therapeutic substances like insulin for the treatment of diabetes are being generated by cloning techniques. So just have a basic understanding of what this technique is all about. So what we have on the upper right-hand side of this figure is these are circles shown in the magenta, and what these represent is vectors containing human genes. So in the magenta, the circle in the magenta represents some sort of bacterial vector, and these little colored rectangles represents some human DNA insert. For example, let's assume that this vector here has a red rectangle. Let's assume that red rectangle represents the insulin gene that has been inserted into this vector. And let's assume in the green rectangle represents the beta globin gene that has been inserted in this particular vector. And let's assume these other vectors, this is another vector here that has spacer DNA or junk DNA or whatever. So this represents 3 vectors containing 3 segments of DNA, but you got to remember there's going to be hundreds if not thousands of vectors. Each vector has a different human DNA insert, either genomic DNA or cDNAs. So what's done after these vectors with the human genes or human DNAs have been generated, what's done is to then have a culture of bacteria. So what these ovals represent are a variety of bacteria like E. coli, and then what's done is to take the E. coli and then add to the E. coli these vectors containing the human genes. Now watch what's going to happen when we take bacteria and add to them the vectors containing the human genes. And in a process called transformation, also called transfection, notice what's going to happen. What's going to happen is E. coli is going to take up a different vector containing a different human DNA insert. Let's assume that this E. coli right here picked up the vector that contains the human insulin gene. And this E. coli picked up a vector containing the beta globin gene. And this is an E. coli that picked up a vector that contains another gene or another human DNA insert like spacer DNA or some junk DNA or whatever. Most E. coli don't pick up any vector at all, and the reason is transformation, that is transfection, is a rare event. So the first thing we want to do in the cloning process is get rid of all those E. coli, all those bacteria that did not pick up any vector at all. And the way it's done is, so now we have a culture of E. coli. Most E. coli didn't pick up any vector at all. Some E. coli did pick up a vector containing a human DNA insert. So what's done is to get rid of all those E. coli that did not pick up a vector at all. And the way that's done is in this culture, what's added is some sort of antibiotic. In this example here, we add ampicillin. Now what's going to happen is, you know from microbiology, ampicillin will get inside the E. coli, and the ampicillin then will kill the bacteria that did not pick up any vector at all. So those bacteria that did not pick up a vector will be killed, and eventually they disappear. But the E. coli that did pick up a vector, they survive. Now, why did they survive in the presence of ampicillin? And the reason being, remember, way back when we talked about a replication vector. The replication vector, the third criteria is they have to have a gene that encodes for an enzyme which inactivates an antibiotic. In this case here, the antibiotic is ampicillin. So these E. coli that picked up a vector has a gene which inactivates ampicillin. In other words, they survive in the presence of this antibiotic. So, but the important feature is we got rid of a lot of E. coli that didn't pick up any vector at all. The next thing that happens in the cloning process is to take the bacteria that did survive in the presence of ampicillin because they have a vector in them. Again, what we're trying to do here is clone the insulin gene in this example. So what's done next is, again, the technique that you have all done in microbiology. You take this mixture of bacteria that survive and then plate them on soft agar dishes. And then anywhere where a single cell sat down on the soft agar, a colony will now grow. You come back the next day, what you will see on this agar dish is all these little white specks on the agar dish. Each speck represents a colony which originated from a single cell. But let's assume this is the E. coli right here. This is E. coli that sat down on this piece of the agar dish and sat down right there and formed colony. So this is a colony of E. coli that harbors the insulin gene. And let's assume that this E. coli, which has the beta globin gene, sat down right here on the agar dish and formed a colony. So this is E. coli that harbors the beta globin gene. But again, what you see the next day when you come back, you see all these little white specks on the agar dish, and the question becomes, how do you know which colony has the insulin gene? All you see is white specks, but you know that at least one of those colonies has the insulin gene. How do you detect the colony that has the insulin gene? So what's done is to take this agar dish with each of these little specks of colonies all over the plate. What's done is to take a little piece of paper. And what's done is take this piece of paper and then blot the paper with, blot the agar dish with a piece of paper. That's called replica plating. And then you take the piece of paper away. Importantly, a representative of each of these colonies now appear on this piece of paper at the right position. So again, this is what this represents here is a piece of paper which was blotted on the agar dish. A representative of each of these colonies now appears on this piece of paper. So again, a representative of this colony now appears on this piece of paper at this position. By the way, this entire plate, as we talked about earlier, is called a DNA library. But again, let's assume a representative of this colony sat down on this piece of paper. Then what's going to happen is this is the colony. This is a representative of that colony which harbors the insulin gene. And let's assume a representative of this colony is now appears on a piece of paper, and this is the representative that has the beta globin gene. And all these other representatives have different segments, different DNA, human DNA that were in the original vectors. Again, what we're interested in is the representative that harbors the insulin gene. How are we going to detect them? What's done is to take this piece of paper, add it into some sort of flask or some dish, and then add a solution which contains some sort of detergent. And what's going to happen is the detergent will then lyse all of the bacteria. And now all of the DNA inside the bacteria is now exposed. So the bacteria are lysed, and then we take those lysed bacteria and then add another solution to the lysed bacteria, and that solution now importantly has a probe which is complementary to the gene of interest. What's the gene of interest here? It's the insulin gene. So we can make a probe in a laboratory or make a probe or commercially purchase the probe that's complementary to the insulin gene. And the probe is made radioactive, and most often the radioactivity is due to radioactive P32. So what's going to happen is when we take this piece of paper with all the lysed bacteria, the probe in solution, the probe will look at all of the DNA in all of those lysed bacteria, looking for the insulin gene in order to base pair with it. When it finds the insulin gene, the insulin probe, the insulin radioactive probe, then will base pair with the insulin gene. But it won't base pair with any other gene because it's not complementary to the other genes or other DNAs. So now, what happens? The radioactive probe is not, has no color, so you still can't see anything. Even though you have the probe, you added it to the DNA, you know that somewhere along the line, hybridization took place. The insulin probe found the insulin gene. Now, how are you going to detect it? So what happens is you take this piece of paper, you wash away all the excess probe, you take the piece of paper, you dry it, and now overlay it with X-ray film. Anywhere where the radioactive insulin probe hybridizes with the insulin gene, what will happen is a dark spot will now appear on the X-ray film. And then because of some sort of grid network that we have now put onto this piece of paper, we know the exact position of that dark spot. And then we go back to the original colony, the original agar dish, and because of the grid network, we know the exact position of that colony. So again, just by the grid network, we know where that dark spot is here. We know that way back on the agar dish, this is a colony which now harbors the insulin gene. And now all we got to do is take a little bit of this colony, and if you grow up lots of that bacteria, you put it into some growth medium, grow up lots of that colony, we have now have E. coli. We have now cloned the insulin gene because E. coli, as it grows, is now making tremendous numbers of the insulin gene. But it's not making the insulin protein if a replication vector is used in this type of technique. So So you have large amounts of the insulin gene, not the insulin protein, if a replication vector is used. But you might say, well, I want the insulin protein to help out with diabetes. I don't want the insulin gene, I want insulin protein. Well, if you want the insulin protein, you would go through the same type of technique as you see over here. Except at this stage over here, what's done after the bacteria are lysed with detergent, what's done is to add some sort of radioactive antibody that's going to see and bind to the protein of interest. What's the protein of interest? It's the insulin protein. So if we have an antibody, an iodinated antibody that will bind to the insulin protein, and then after washing away excess radioactive antibody, we take the piece of paper, overlay it with X-ray film. The radioactive antibody will then strike the X-ray film. A dark spot will now become evident. Then you go back to the agar dish. You take a little bit of that colony and grow it up. And now that E. coli is making large amounts of the insulin protein. But the most important feature is over here. Again, this is all being done by the grid network. But the most important feature over here, what kind of vector is being used in order to generate insulin protein? It has to be an expression vector, not a replication vector. It has to be an expression vector. And what were the two additional criteria that must be met? The expression vector must have engineered into it a promoter to which the RNA polymerase of bacteria must recognize, as well as a Scheindel-Garner sequence must be engineered so that the bacterial ribosomes must therefore recognize and then jump onto the RNA in order to make insulin protein. Now, this type of technology, by using expression vectors, is widely used now in medicine for generation of many therapeutic substances like insulin for helping out with diabetes. In the old days, if someone had diabetes and you want to help and give them insulin, that insulin came from pigs. They had to sacrifice a multitude of pigs in order to get a little bit of insulin to help out with diabetes. But nowadays, insulin is generated by cloning technology. Also, in hemophilia, a lot of the blood clotting factors like factor 8, factor 9 are being generated by cloning techniques. And a lot of vaccines nowadays are being generated by cloning techniques, such as the hepatitis B surface antigen being generated by this type of expression cloning techniques. So it's obvious why someone would want to use an expression vector in order to get a recombinant protein. But why would you ever want to get large amounts of the gene, shown here on the left side? Why would you want the gene and not the protein? Well, the answer why you want the gene is for gene therapy as well as for the preparation of transgenic animals. That's what's coming up next. So on this figure here is the use of that cloned gene that we talked about on the previous page. So we have large amounts of a gene that we have now cloned, and what's shown here is retroviral gene therapy. Now, you know from microbiology, retroviruses have 3 genes, and the genes encode for the GAG protein, for the Pol proteins, the Pol proteins being reverse transcriptase as well as integrase. As well, there'll be a gene for the envelope protein. So what's done in retrovirus gene therapy is a portion of the viral genome is taken out and replaced with that cloned therapeutic gene that we obtained on the previous page. So you take out a little bit of the viral genome, you replace it with this cloned gene. The important feature is here, the virus now is called a replication defective retrovirus. It cannot reproduce its own genome because why? Because a portion of it was removed and replaced with this therapeutic gene. But the important feature is, now that we've taken out part of the genome of the virus and replaced it with a therapeutic gene, the virus now thinks that therapeutic gene is part of its own genome. So the inserted gene is now part of the viral genome. The virus thinks that new gene is part of itself. What's done now is to take this replication defective retrovirus, it's then packaged. And once it's packaged, it's then added to some sort of target human cell. What's going to happen is the virus then will insert itself into that target cell. And once it inserts itself into that cell, what's going to happen is that virus can express its genes into proteins. Even though the virus cannot reproduce itself, it can still express its genes into proteins. And one of its genes, remember, is that therapeutic gene. And that gene will be converted into messenger RNA, which will now be made into protein. So now, once that replication defective retrovirus gets into a target cell, it expresses its genes, and one of its genes is the reverse transcriptase. and reverse transcriptase will then take the RNA and convert it to DNA. And one of its genes is in the Pol gene is the integrase, which will then take the DNA and integrate the DNA into the chromosomes. And then once it gets into the chromosomes, it continues to express its genes in the proteins. And again, one of its new genes is that therapeutic gene, which is now being made into protein by first making it into messenger RNA and then made into proteins. So this is how a retrovirus is modified and then be used to make a recombinant protein. Now, how was this replication defective virus used clinically? Now, that's shown here in this ex vivo replacement therapy slide. Now, what is ex vivo replacement therapy? What that stands for is that cells are taken out of a human, and then when they're taken out of a human, those cells are then manipulated. You add back you add to those cells this therapeutic gene, and then to these cells, and then these cells have been altered, and then you give them back to this human being, and the idea being trying to cure a defective tissue. So what we have here is some human in which CD34 plus cells have been taken out of this individual, and then these CD34 plus cells are then placed on some sort of dish. And then in the meantime, we have generated a replication defective retrovirus, which in this example, we have taken out a piece of the retrovirus and replaced it with the clone gene for interleukin receptor gamma chain gene. So this is a retrovirus which cannot reproduce itself, but still can express its genes into proteins. So now this replication defective retrovirus is packaged, and then when it's added to those CD34 plus bone marrow cells, what's going to happen is those virions then will invade the CD34 plus cells, and then importantly, the therapeutic gene integrates along with the rest of the viral genome, and importantly, that therapeutic gene now is being expressed into messenger RNA and then being expressed into protein. In this example here, into the protein interleukin receptor gamma chain protein. And then those altered cells are then put back in the human being, and the idea is that you're trying to cure a defective tissue, because now therapeutic gene is being made into recombinant protein and you're curing a defective tissue. Now, this type of technology is being tried for certain diseases such as severe combined immunodeficiency, in which the interleukin receptor gamma chain gene has been mutated, causing SCID, or other situations where adenosine deaminase, ADA deficiency, where the gene has been mutated. So again, normal adenosine deaminase has been cloned. It's been put back into replication defective retroviruses and put back into SCID patients, with the idea, hoping that the good ADA, normal ADA protein will be made and helping out by curing a defective tissue. In addition, this type of technology is being tried with cystic fibrosis, with trying to replace a defective chloride channel protein with good normal chloride channel protein. And in another example where ex vivo replacement therapy has been tried is with severe hyperammonemia, in which there's a defective ornithine transcarbamylase gene giving rise to severe hyperammonemia. Now, there's problems with this type of ex vivo replacement therapy. Even though it sounds very good on paper, there's some major problems with using this type of replacement therapy. And one is the problems with this type of gene therapy is targeting the right tissue. Even though you take back these cells, which have been manipulated, and put them back into a human individual, how can we guarantee that those cells go to the right tissue? That's a serious problem, is targeting of the cells going back to the right tissue. Another problem with this type of gene therapy is integration of the gene into the chromosomes. How do we know that the replication defective virus integrates itself properly into 46 chromosomes? That's another serious issue in this type of replacement therapy. Another problem with this type of gene therapy is activating the gene. Sure, we can get integration of the viral genome into the chromosomes, but then how do we get proper expression of the gene, in the therapeutic gene, into protein? We don't want to make too much or too little. We have to activate to make the proper amounts of the protein in order not to swamp the tissue with too much protein. So that's another serious issue, is activating the gene into messenger RNA, into protein in the right amounts. And lastly, another problem with this type of gene therapy is avoiding harmful side effects. Some of these types of replacement therapies have resulted in toxic situations occurring, as well as immune responses have been a major problem. To illustrate this, a few years ago you might have heard about at the University of Pennsylvania in Philadelphia, there was a patient who had severe hyperammonemia. He was a 21, 22-year-old male who had severe hyperammonemia. Why? Because he had a defective ornithine transcarbamylase. And so the normal ornithine transcarbamylase gene was cloned. It was put into a replication defective adenovirus with the idea that the adenovirus would go to the liver because that's where the enzyme is located. Ornithine transcarbamylase is part of the urea cycle, which is in the liver only. So by putting it into a replication defective adenovirus, the idea being the adenovirus would go to the liver, it would express the genes into proteins, and hopefully the ornithine transcarbamylase, which is now part of the adenovirus genome, would be expressed into correct, normal ornithine transcarbamylase, and therefore cure the hyperammonemia. Unfortunately for this individual, it did not work. This person had a lot of immunological problems as a result of this ornithine transcarbamylase, and unfortunately he died shortly thereafter. And then it was written up in magazines and newspapers, and it set back gene therapy pretty severely because of this adverse effect that happened because of the immunological response due to this type of therapy. But unfortunately for this individual, it's just simply a technical issue which hopefully can be rectified in the near future. So this is ex vivo replacement therapy. Now another type of use of cloned genes, and I'm going to stress, by the way, that this is somatic cell gene replacement. And the important feature is here, why is this being done in humans? And the reason is this is somatic cell gene replacement, and importantly, these genes that are being inserted back into humans, they are not inherited. There's no ethical issues involved here, that these genes that are being added, they're being added to a single individual in somatic cells, so that gene is not inherited. But now what we're going into is an idea where genes can be inherited, and that's involved in the construction of transgenic animals. So here, that gene that we can clone based on procedures we talked about earlier, we can now take the cloned gene and put it into a germline. Now this would be referred to as germline gene replacement. The issue is here, because the gene is put into a germline, the genes can be inherited. And that word inherited now brings up a major ethical issue. So with human beings, at least overtly, this is not being done. But where this is being done is in mice and other animals in order to construct transgenic animals. Now there's two types of technologies that are involved in generations of transgenic animals, and one is to, as you see in the middle of the page here, is to introduce that cloned gene that we've cloned on the, by techniques talked about earlier. We can take the cloned gene and put it into fertilized ova. In addition, we can take the cloned gene and introduce it into embryonic stem cells. So the first thing I wanted to discuss is the technology behind taking a cloned gene and put it into fertilized ova. Now in your book, what you see is a little paragraph here, but what I'd like to do here is to illustrate this diagrammatically. So what we have here is some recently fertilized mouse. So what's done is to take out the fertilized ova from that mouse and then microinject the cloned DNA. Again, the cloned DNA came from cloning technologies that we talked about earlier. So when we microinject the cloned DNA into the fertilized ova, we then take that ova and put it back into a foster mother, some surrogate mother, then wait a few weeks. After a few weeks, that surrogate mother now will have all the progeny. The important feature is that in the progeny, the offspring are now considered transgenic animals. And that new gene that was put into the ova is now called a transgene. That all of these progeny will have a new gene, which is that transgene. Now what would happen to all of these progeny when they breed with one another? What would happen to the transgene? Would it be inherited or not? Well, the answer is when these progeny with the transgene in them, when they interbreed, the transgene would be inherited. Why? Because the gene was inserted way back over here in the ova, in the germline. So the transgene is indeed inherited. Now this type of technology is being used for the generation of transgenic animals. And for example, if you wanted to study, let's assume you had some new drug, some new statin drug for the treatment of hypercholesterolemia. One thing you do not want to do initially is take the drug and test it with humans. So you would rather test it with animals which have hypercholesterolemia. So one of the ways this could be done would be to get a defective gene for the LDL receptor, take the defective gene for the LDL receptor, clone it, and then insert it into ova, as you see over here, and then take the ova, put it back into a mouse, and all of the progeny from that mice now would have a defective LDL receptor. The consequence is all of these progeny now would have hypercholesterolemia, and you can test the effect of some sort of statin drug that way. So this is a generation of models for human diseases. But this type of technology does have problems, and one of the problems with this type of technology is there's no control of the integration site. If you take a gene and you put it into a fertilized ova, where does that gene insert into chromosomes? Into a mouse. So controlling where that gene goes is a problem. Another serious problem with this type of technology is the expression of the gene into messenger RNA and then downstream into protein can be erratic. Sometimes you might get too much and sometimes you might get too little protein being expressed. So that's a serious problem with this type of technology. But the best type of technology for generation of transgenic animals is shown on the last figure of chapter 6. And that has to do with taking the clone gene and to put it into embryonic stem cells. So there is a figure in your book, which is the last figure on chapter 6. This is the constructing of transgenic or knockout mice. So what's done in this type of technology is take some recently fertilized mouse and take out the blastocyst from that mouse, and that's shown over here. So there's a multitude of cells in this blastocyst. And then we take this blastocyst and then convert the blastocyst into single cells. And we take the single cells and put them into some sort of dish. So here we have the single cells from the blastocyst sitting in a plate. And then we add to the cells that clone gene, any clone gene that we clone from technologies talked about earlier. We take the clone gene and then add it into the solution, and then the gene will be taken up by those single cells from the blastocyst. And importantly, homologous recombination will take place, in which that gene will integrate into a particular site of a chromosome as a single copy. So that gene, by homologous recombination, integrates as a single copy at a specific site at a specific chromosome. Now, how is that done? That's all details that you don't have to know. But importantly, you're taking a gene and inserting it at a specific site in a single copy. So what else can be done with these embryonic stem cells, these single cells? What can also be done is to add some sort of foreign DNA which will insert in a particular gene. When this foreign DNA inserts into a particular gene, what happens to that gene? It's no longer functional. It's defective. In other words, you have knocked out that gene. It can no longer be expressed into messenger RNA as well as into protein. And that's what's called knocking out a gene. So notice how two diverse situations. One, you insert a new gene, and the other, you have knocked out a gene in embryonic stem cells. So now we have a plate. Let's assume we have inserted a new gene. So we have a plate of blastocyst cells. We inserted a new gene. And what's done, these embryonic stem cells can differentiate into all types of cells when introduced into another embryo. Now, where is this other embryo coming from? Over here on the upper left-hand portion of the figure, here's another mouse, a recently fertilized mouse. You wait a short period of time, and now we have another blastocyst here. And what's going to happen is we now take these cells, these single cells, these altered embryonic stem cells. If these are then introduced into this second blastocyst, what's going to happen is we will now generate what's known as a chimeric blastocyst, in which the blastocyst now has a mixture of cells. Some cells came from this mouse over here with a light-colored blastocyst, and the darker cells coming from the darker colored blastocyst shown here on the right. So this is a chimeric blastocyst, which consists, it's a mixture of cells coming from two blastocysts. Now, we take this chimeric blastocyst, it's then infused into some sort of surrogate mother or foster mother, and then we wait a few weeks, and then what's going to happen to that mother? We'll have all these progeny. Now the progeny now are called chimeric offspring. The progeny now will have all these patches throughout the body. Why do they have patches? And the reason why they have patches is because they have that the blastocyst, the original blastocyst, has two kinds of cells. Some were light colored cells, which gives rise to light colored patches on the mouse. Some of the darker colored blastocyst cells gives rise to dark patches on this mouse. So this is a mixture of cells from this blastocyst giving rise to a chimeric mouse with all these different patches that it has in them. Also importantly, some of these embryonic stem cells can get into the germline, and when they get into the germline, importantly, we can now we can generate transgenic sperm. When they fertilize a normal egg, downstream from that, we can get a transgenic mouse. And the issue is now all cells in that transgenic mouse now will harbor that trans gene. Every cell in the body, there won't be patches, but every cell in the body now will have that trans gene. Now, where is this type of technology being used? This type of technology that is being used is for studying gene function and gene regulation. If we wanted to study, again, the LDL receptor function, we can get a normal gene for LDL receptor or a defective LDL receptor gene and analyze the function that way. Another example is for analyzing or studying model diseases. Again, another good example of this is for Alzheimer's disease. And so this type of technology it's only in its infancy, but as time goes on, it'll be a very dominant type of technique that's used for studying human disease in an animal model.